We're actually fighting a fascinating, completely invisible war every single day. I mean, we are vastly outnumbered by bacteria, viruses, and spores, but humanity has this secret foundational weapon that we use to keep them in check. Temperature. By manipulating heat, we literally manipulate the survival of these invisible organisms. Welcome to this explainer. Let's dive right in. Picture this. It's the 1920s. A young British doctor is desperately fighting a deadly typhoid fever outbreak in a small Welsh coal mining town. When his very first patient dies, he realizes, okay, I have to break this chain of infection right now. So what's his solution? He takes the patient's bed sheets, all their clothing, their personal effects, and just tosses the whole shebang into a massive, roaring bonfire. It's a dramatic scene, but it brilliantly captures our most primitive, extreme, and undeniable weapon against disease: raw fire. Now, that bonfire represents incineration. It's an extreme form of physical microbial control that uses a direct flame to turn contaminated materials entirely to ash, killing microbes incredibly fast. It is the absolute, ultimate brute force purifier. And we don't just see this in 1920s novels either. Think back to 2001, when a devastating foot and mouth disease outbreak hit British cattle. Authorities quickly realized that standard disinfection just wasn't going to cut it. To stop the spread of this highly contagious virus, they had to order the mass incineration of nearly 4 million hoofed animals. 4 million. It's a really harsh reality, but sometimes raw flame is literally the only absolute guarantee of destruction. So, here's our blueprint for today's explainer. We're going to start with the ultimate purifier, fire. Then we'll look at the actual science of microbial death, explore the clash between dry and moist heat, check out the gold standard of autoclaves, and finally, wrap up with pasteurization in everyday life.
All right, part one, the ultimate purifier, fire.
Okay, so we've talked about these massive, dramatic bonfires, but let's scale that immense destructive power down a bit. Let's look at how direct flame is actually used as an everyday, rapid sterilization tool. If you've ever set foot in a biology lab, you definitely recognize this glowing blue flame, right? The Bunsen burner. Scientists harness this extreme heat every single day. Before a microbiologist takes a bacterial sample, they just pop their metal inoculation loop right into this flame for a few seconds. That direct flame burns away any rogue organisms, dust, or lint instantly. It's the exact same brute force principle as the bonfire, just highly controlled and localized to make sure the lab stays totally sterile.
Moving on to part two, the science of microbial death.
Now, obviously, we can't just set absolutely everything on fire. That'd be chaos. We need to be way more precise. And here's something really interesting. When we heat up a bacterial population, they don't all just drop dead in the exact same millisecond. Actually, microbial death happens exponentially. Food scientists and microbiologists measure this using something called the decimal reduction time, or the d-value. Simply put, this represents the exact amount of time it takes, at a very specific temperature, to kill exactly 90% of the living organisms in a sample. It's a totally crucial metric because it proves to us that this death is a constant, predictable rate, not just some sudden random event. You can really see how precise this math is in action on this chart. Let's say we start with a massive population of 1 million cells at minute 0. Well, after 10 minutes of heating, 90% are wiped out, leaving 100,000. 10 more minutes, another 90% die, leaving 10,000. It's kind of like a giant puzzle coming apart, piece by piece. This shows us that with enough time and the right consistent temperature, an overwhelming million microbes can be methodically reduced to just one single surviving cell over the course of 60 minutes. It's just pure, predictable math.
All right, part three, dry versus moist heat.
So we know that heat kills over time, for sure. But the way we actually deliver that thermal energy, that makes a huge difference. Think of dry heat, kind of like your kitchen oven. It's essentially baking. It slowly burns or oxidizes the microbes. But here's the catch. Air is a terrible conductor of heat. Because it has such poor penetration, you literally have to blast materials at super high temperatures. We're talking 160 degrees Celsius for up to 2 hours just to destroy resistant bacterial spores. Now, moist heat, on the other hand, is equivalent to boiling. Water molecules conduct heat way, way better than air. Because of this, moist heat rapidly denatures and coagulates the proteins right inside the bacteria. It requires less heat and less time overall because the thermal penetration is just incredibly efficient.
But wait, even though moist heat is definitely superior, simple boiling water isn't actually a perfect sterilizer.
Yes, most standard pathogens are going to die within about 10 minutes in boiling water at 100 degrees Celsius. However, bacterial spores, which are basically these super tough, dormant survival structures of certain bacteria, they can easily survive boiling water for 2 whole hours or even more. No way, right? So if we need true 100% sterilization, especially in places like medicine, we clearly need a much heavier hitter than just a pot of boiling water. Which brings us to part 4. Autoclaves, the gold standard. If we're going for absolute sterilization, we absolutely have to have a device that definitively wipes out those stubborn spores. Let's look at the evolution here. Back in 1680, on the left, Denis Papin invented a steam digester, which was basically the world's very first pressure cooker. Fast forward to 1880, and Louis Pasteur's collaborator, Charles Chamberlain, adapted that design into a pressurized steam sterilizer. He used it to prove that really high heat could defeat the theory of spontaneous generation. And on the right, you see the direct descendant of that invention, the highly regulated, heavily engineered modern autoclave. We use this all over the world today to completely sterilize blankets, surgical instruments, and lab media. Let's quickly walk through this mechanical process step by step, just so you can see exactly how this marvel works. First, the contaminated articles are placed right into the sealed chamber. Next, steam flows into an outer jacket, and that forces all the cool air out of the main chamber. Then, a special valve completely seals the system and cranks the pressure up to 15 pounds per square inch above normal atmospheric pressure. Now, because the steam is trapped under all this high pressure, it doesn't just boil at 100 degrees Celsius, it superheats to an incredible 121.5 degrees Celsius. This superheated environment literally annihilates even the most resistant bacterial spores in just about 15 minutes. Now, there's a really crucial point we need to clarify here because there's a very common misconception. When people think about autoclaves, they almost always assume that the immense physical pressure is what's actually crushing and killing the bacteria. Actually, scratch that. That is absolutely not the case. The pressure is strictly a mechanical trick. It merely forces the steam to reach that critical temperature of 121.5 degrees without just boiling away. The true sterilizing agent, it's the moist heat itself. It's those highly energized, super hot water molecules that actually penetrate the bacterial cells, coagulate their proteins, and take them out. And finally, part 5, pasteurization in everyday life. Okay, let's bring the science out of the hospital operating room and right into your own kitchen refrigerator. We just talked about the incredible absolute power of the autoclave. But think about it. We cannot autoclave milk or fruit juice. If we exposed milk to superheated pressurized steam at 121.5 degrees, it would curdle, caramelize, and completely ruin both the taste and the nutritional value. It'd be gross. So we need a totally different approach for our food. And for this, we rely on pasteurization. It's a gentle, highly precise application of heat that's specifically designed to reduce the microbial population without destroying the liquid itself. The public health benefits of this method are, honestly, absolutely massive. Pasteurization safely safeguards our milk and our fruit juices. For decades, its main target was actually destroying Mycobacterium tuberculosis, which prevented massive, deadly outbreaks of TB spreading through dairy. Today, it's carefully calibrated to destroy Coxiella burnetii, which is a highly heat-resistant pathogen that causes Q fever. It also easily eliminates common foodborne villains like Salmonella and E. coli. But remember the really crucial distinction here. Pasteurization strictly reduces the pathogenic population. It does not achieve total sterilization. Because those stubborn spores survive, total sterilization isn't reached. Which, by the way, is exactly why your milk eventually goes bad, even when it's just sitting perfectly in the fridge. So we've journeyed from raw, dramatic bonfires to the precise mathematics of destruction, through high-pressure autoclaves, all the way to the pasteurized juice sitting in your fridge. It really is a remarkable, invisible war that we win daily through the power of temperature. But I want to leave you with this final, somewhat provocative thought to ponder. If microbes suddenly developed a universal immunity to our ultimate weapon of heat, what would happen to our global food supply, our modern hospitals, and ultimately our survival? It really makes you look at a simple flame or a boiling pot of water in a completely different light, doesn't it? Thank you so much for joining me on this explainer. Keep learning, and I'll catch you next time.